Welcome to my YouTube channel. Please be sure to subscribe to my channel and also do not forget to press the bell icon so you never miss any video tutorial published by me. Welcome back to my YouTube channel Conquest Physiology. So students, today let us discuss two pathways of blood clotting, intrinsic pathway and extrinsic pathway. Before that, let us discuss about blood, composition of blood, right? So the blood in our body, which is known as soft tissue, comprises two parts: plasma part, which is known as liquid part. It is in 55% in our body. Formed elements or blood cells, they are in 45% total. Like corpuscles, red blood corpuscles, white blood corpuscles and platelets. They are in 45%. Now, out of this 55% part, 90 to 92% is water, and it has 6 components basically, and plasma proteins are 6 to 7%. Right. So plasma proteins are of 3 types: albumin, globulin, which is also of 3 types: alpha globulin, beta globulin and gamma globulin, and fibrinogen is 3rd type of plasma protein. So these are 7%, out of which 4% are 4% plasma protein is albumin, 2% plasma protein is globulin, and 1% is fibrinogen. Right? And the 3rd component of plasma part of blood are ions like calcium, sodium, bicarbonate ions, chloride ions. 4th part is gases like oxygen, carbon dioxide, nitrogen, oxygen, carbon dioxide, nitrogen, etc. 5th component of plasma part of blood is NPN substances, non-protein nitrogenous substances, which include urea, creatinine, etc. And 6th part is nutrients. Now that was about liquid part of blood. Apart from liquid part, blood also has corpuscles, which comprise 45% portion of blood. There are 3 types of cells: red blood cells, also known as erythrocytes, which are 99%. Out of this 45%, 99% are red blood cells, and their main purpose is to transport oxygen in the body. White blood corpuscles or WBC or leukocytes, they comprise 0.2%, and their purpose is defense or provide immunity to our body. 3rd part is platelets or thrombocytes, which are 0.8% out of the total 45%. Their purpose is blood clotting. But these platelets or thrombocytes do not perform blood clotting without 13 clotting factors. These 13 clotting factors are type of proteins, and they all are important for blood clotting. Like, even if one of these factors is absent in the blood, then blood clotting will not occur. So all of these 13 clotting factors are important for blood clotting. So now let us discuss some important terms related to blood clotting and blood functions in our body. So students, now let us know the names and the way to remember the 13 clotting factors which participate in blood clotting. The first factor, these numbers in Roman, they are indicating their chronological order of when we found them. Like fibrinogen was found first, so it has been named as first. It doesn't mean that in clotting, during clotting, it will be used first. It is just showing the order of its discovery, the Roman number. So first factor to be discovered was fibrinogen. Second was prothrombin. Third, thromboplastin. Fourth, calcium. Proacylarin. Fifth, proacylarin, which is also known as labile factor. Sixth is acylarin. Seventh is stable factor. Eighth is anti-hemophilic factor. Ninth is Christmas factor. 10th is Stewart-Prover factor. 11th is plasma thromboplastin antecedent. 12th is Hagman factor, a glass factor, and 13th is fibrin stabilizing factor. That is also 12th factor is also known as glass factor. Hagman factor is same as glass factor, and the 13th factor is also known as Laurent factor. Now the mnemonic device to remember these factors is like for permanent treatment of is like there is no factor like this. For permanent treatment of cancer, please arrive soon at Chandrasekhar Private Hospital, Faridabad. So F is for fibrinogen, and similarly all these, the first letter of each word is indicating the factor associated. So that's how we can easily remember these factors. So now let us discuss the intrinsic and extrinsic pathway and when they are activated. So students, now let us understand the definition of blood clotting. Blood clotting is basically a cascading mechanism which requires a lot of enzymatic reactions. The process of blood clotting is quite complex, which requires all the 13 factors as I have already discussed, and they all act in a specific cascading sequence, which results into fibrin meshwork, and ultimately this fibrin meshwork forms a stable clot. So that's about blood clotting. Now let me talk about two conditions which are antagonistic. Thrombosis is a condition when clotting in an undamaged blood vessel occurs. Usually in blood vessels, blood is in liquid form, like there is no clot. But during thrombosis, a clot is formed, and the blood vessels are also not damaged. At that time, a clot is formed. In case the blood vessels are undamaged, then the condition is known as thrombosis. In contrast to this, hemorrhage is a process when blood takes too long to form clot. Like blood will keep on oozing out of the blood vessels because clot will not form. So obviously blood will keep on coming out of the blood vessels. Now there are two pathways of blood clotting. The first one is intrinsic pathway and the second one is extrinsic pathway. So when there is a trauma in the blood, then intrinsic pathway is activated, and extrinsic pathway when tissue trauma occurs, like very severe kind of trauma occurs, that time extrinsic pathway comes into picture. Now the second difference between both of them is intrinsic pathway takes very long time to form the clot, like it may take around 4 to 5 minutes, whereas extrinsic pathway takes around seconds, a few seconds. It occurs rapidly within seconds. So the third one is activators, which activate the cascading mechanism by acting upon the clotting factors. They are present, they are either in the direct contact of the blood or they are intrinsic to the blood, like they are within the blood. But in extrinsic pathway, the activators arise from outside, like it leaks into the blood vessel to mix with blood. So they arise from outside cells, like from some other tissues. And the fourth difference is it has more steps, whereas extrinsic pathway has less steps. So now let us discuss extrinsic and intrinsic pathway in detail, like how it has more steps, intrinsic pathway, and how extrinsic pathway has less steps. So let us discuss their mechanism of action in detail. So now let us understand the mechanism of blood clotting via intrinsic pathway and extrinsic pathway. First, let us understand intrinsic pathway. Basically, these factors, when they are not showing small a as a subscript, this form is activated form and this is inactivated form. These are like proteins, so they keep on rotating with the blood in inactive form. In inactive form, they are known as zymosins. Once zymosins are cleaved and some part of zymosin is removed, then it becomes activated enzyme. So let us understand how exactly it occurs. So there are some negatively charged molecules, factors like kallikrein and HMKW. The full form for it is high molecular weight kininogen. These are negatively charged. So this one is a serine protease, like it is an enzyme. So it will cleave this zymogenic form of factor 12 into activated form. It will cleave some part of zymogen factor 12 and it will become activated to form 12A. Now what 12A, activated form of factor 12, will do is it will activate factor 11. Factor 11 is also in zymogenic form. So once activated form of 12A will act on it and it will become 11A. That means it will also become activated. Now 11A will further activate factor 9 and factor 9 will become 9A. Like it will also become, it was also in zymogenic form, now it will become activated. And then after becoming activated, that is 9A, it will form a complex with factor 8 and this complex will require calcium. Calcium will stabilize some part of these molecules, these proteinaceous molecules, macromolecules. So calcium is required, calcium which is the fourth factor. So like it will stabilize the complex of 9 and 8. 8 will also become activated from its zymogenic form. Now along with PP, that means plasma phospholipids, these are also present in blood. They all will form complex 9A, calcium, 8A and PP, and the entire complex will act on factor 10th. Now this factor 10th will also become activated from its zymogenic form, factor 10A. This factor is very important and it is important in both the pathways. Like extrinsic pathway will also lead to activation of factor 10A. We will understand extrinsic pathway also. First of all, let us understand the entire intrinsic pathway. So factor 10 will become 10A from its zymogenic form, and along with calcium and factor 5, it will, like calcium is again required for the stabilization and catalyzing some reactions. So now all these three will act upon prothrombin, which is factor 2. And prothrombin is zymogenic form, and once some part of it will be cleaved, then it will become thrombin, which is activated part of factor 2A. Now what will happen is thrombin will act on fibrinogen, which is factor 1st, and fibrinogen will become fibrin. Now from here, like it is a monomer, fibrin is a monomer. So fibrin will polymerize. It will form a like long chain, and it which will ultimately lead into fibrin thread. It will form a thread-like structure. And fibrin thread will form loose and friable meshwork with blood cells, like clot type of thing. But it will be loose. Like when we have some injury or something, then we realize that at that time when the blood has just started clotting, at that time it is a loose mass. So the same kind of mass will be formed. Now factor 13th will come into picture, and factor 13 will change into its activated form, factor 13A, and along with the help of calcium it will form a stable clot. Ultimately, after 4 or 5 minutes, we realize that the blood clot becomes very hard. So that is a stable clot. Initially it forms a friable meshwork. So factor 13A basically converts this loose friable meshwork of complex with blood to form a stable clot. So that's how blood clotting occurs. Now let us understand extrinsic pathway as well. In extrinsic pathway, factor 7th along with TF or tissue thromboplastin, which is factor 3rd, it gets activated into factor 7A. Once it becomes activated, then along with thromboplastin, then with the help of thromboplastin and calcium, it acts upon factor 10, and factor A gets converted into 10A, and then the entire procedure is the same. So basically extrinsic pathway has less steps as compared to intrinsic pathway. So that's how blood clot is formed in our body. So if we will count, we have mentioned all the factors except for factor 6, because the role is not clear. Role of factor 6 is not clear, but it has some role. But the research is going on. So till now we do not know what exactly factor 6 does. So it is basically unspecified yet, but we are sure that it is the 13, like it is one of the 13, it is amongst one of the 13 factors and which participate in blood clotting. So if we will see, we have included all the factors 1, 2, 3. Let us count one by one. 1, 2, 3. 4 is calcium. And factor 5, 6 role is unspecified. Factor 7, factor 8, factor 9, factor 10, factor 11, 12 and factor 13. So we have discussed all of them. So that's all about blood clotting. That's how blood clot is formed in our body.